So Ashley's a senior staff scientist with our molecular genetics research development group at IDT. She's been part of IDT for 13 years and has conducted research in RNAi and antisense oligotechnologies, and more recently has been focused on the development of novel CRISPR RNA sequences and modification patterns that allow for more efficient and specific cleavage of the SP-Cas9 and AsCas12a CRISPR nucleases. So with that, I'll leave it to Ashley to go forward.
Thanks. All right, well, I'm excited to talk to you guys today about all the work our R&D team has done to make efficient tools for CRISPR-Cas9 and CRISPR-Cas12a genome editing. And Carl and Sarah both laid the stage nicely to dive into CRISPR now. And so what our R&D team is really focused on is providing the complete workflow to allow you help along the way from designing your guide RNAs. So we have a great bioinformatics R&D team as well that we're able to develop really high throughput screens and then feed this into our bioinformatics team to develop design algorithms similar to what we have for dicer substrates, but we now have that for guide RNAs, so you can come to us. We can do the design for you. We provide all the experiment-ready reagents, so you don't need to design any constructs or develop those. We have the guide RNAs. We have proteins. We focus on RNP delivery at IDT. And then if you're doing HDR and you need a donor construct, we have solutions for introducing single-stranded and double-stranded, and I'll get more into that. And then we also have some great options for doing the analysis of CRISPR, and I'm not going to be able to go into all of this today, but I encourage you to talk to Garrett or I if you have specific questions about anything along this workflow. We can provide help both.
It's using background. I think it's right here.
Okay, let me fix that. All right, is that a little better? Okay, so the two main CRISPR nucleases are the most dominant one first came about with strep pyogenes Cas9. We also...
Sorry, I'm going to fix that right now. It's just holding here. Touching.
Sorry, I'm not noticing.
That one right there. That's good.
Okay. All right. So we've got reagents to support Cas9 as well as Cas12a. And so both of these are RNA-guided endonucleases that will make a double-stranded break in the genomic DNA. And the two most common repair pathways, as we've heard discussed today, are the non-homologous end-joining pathway, where the cell's endogenous machinery will correct the double-stranded break and add in insertions and deletions leading to a knockout, or homology-directed repair, where you provide a donor template that can then go in and make a correction like Sarah spoke to. So to look at these nucleases in a little more detail, the Cas9 system has natively has two RNA molecules, a cRNA, which contains your target-specific region, and a tracer RNA, which is universal. These can anneal together. We can also add chemically synthesize these together by adding a linker loop and make this one RNA molecule. These make a blunt double-stranded break upstream of an NGG protospacer, whereas Cas12a is a single RNA that recognizes a TTTV site. So they both make double-stranded breaks, both guided by RNA, recognize different areas in the genome. And so we've really focused our efforts on developing tools to support both of these nucleases. At IDT, we have our research group also has a good amount of effort put into protein engineering and evolution. So we have 4 nucleases that support both Cas9 and Cas12a, making the double-stranded break. And I want to discuss here some problems that were initially discovered with both of these. So wild-type Cas9, there was reports of, we've seen off-target editing. If you have a sequence that has some similarity at other places in the genome, it can make a unintended off-target. So we've developed a high-fidelity protein. This is exclusive to IDT. And then Cas12a, I'll go into more detail, but when this first came out, the efficiency wasn't as high as Cas9, so we did some protein evolution to improve the efficiency of that, and so we've got a protein that's known as Cas12a Ultra. So to discuss a little more the issues with delivering DNA expression, Cas9, and why we lean towards using RNP. When you deliver RNP, this is going to be directly delivered into the cell, be able to act right away and be rapidly degraded. And so you can see here by a Western blot how the protein enters the cell, is very quickly degraded. However, if you introduce a plasmid into the cell, this has got to be transcribed and translated. This is overexpressed, and there have been a lot of reports of how this leads to off-target editing. And to show some data that we developed in our lab for this, here we're looking at where we've delivered a guide RNA with an expression system. And in this case, we're looking at on-target editing. And we have a technology known as RAMP-seq, which is a targeted amplification sequencing where we're able to multiplex up to 5,000 assays at once. So we're able to look at the on-target site as well as predicted off-target sites. And so what you can see here, the level of editing that we see at the on-target site and all of the editing that we also see at other sites that are unintended. And so simply by switching to RNP delivery, we've now maintained the high level of editing, but now we've really reduced that level of off-target editing simply by delivering this as a protein rather than a DNA expression system. However, the third panel here now is looking at a different guide RNA sequence. So this is delivered as a protein also. But you can see now we are still seeing some levels at some off-target sites that are giving about 10% editing. So that led us to try to develop a protein that would reduce this off-target editing. So in 2018, we published a paper in Nature Medicine that highlights this work where through bacterial mutagenesis screens, we developed a high-fidelity protein that's able to reduce off-target editing while maintaining high on-target editing. And so here's the paper, and there's been a lot of other publications of people looking to produce high-fidelity proteins. The issue we saw with a lot of those published constructs is that they were developed as plasmid systems. So when we purified that protein and then looked to deliver those, they had a great reduction in on-target activity. So the evolution of our protein was done in the context of it being delivered as RNP. And so all the data in this publication, and you can see in the figure here that ours being the final construct or the final protein in green maintains that on-target activity at a much higher level. So you're able to reduce off-target activity while still having a high level of cutting. So it's a protein that's exclusive to IDT that we've developed. And so looking back at this same site that I just showed you when we were delivering the wild type Cas9, we still saw a decent amount of off-target editing. So now that we've switched and delivered our high-fidelity Cas9, the on-target editing is actually identical at this site, but we've now reduced all of the majority of all of that off-target editing. So then the other nuclease I spoke to was Cas12a. And so this was discovered sometime after the Cas9. And when the first testing of all of this and we purified this protein, it just didn't work quite as well as Cas9. So we went through a similar bacterial mutagenesis approach to try to evolve this now to have enhanced activity, where our efforts with Cas9 were to try to evolve a protein that had greater specificity. Now we wanted to get Cas12a working as well as Cas9. So we were successfully able to do that, and we have a protein that IDT has developed that's known as Cas12a Ultra. And so this data here shows that we've also done a lot of work in our R&D team to really optimize the protein engineering. So optimizing the NLS sequence and the tag and the structure of our protein. And so the first version of proteins before we had done that optimization was referred to as our V1 proteins. And you can see that the Cas12a, this is an average of about 100 sites, that the overall editing is quite low. Simply improving the protein engineering, we've now brought that level up in yellow to be very effective. And then in green is now our Cas12a Ultra, where if we randomly look at, in this case, 100 different sites, we are getting very high levels of editing, similar to how Cas9 is able to make these double-stranded breaks. So then to compare this to Cas9, here we're looking at 100 sites that are in similar genomic regions. We're comparing in blue the wild-type Cas12a. You can see the range of editing that we see now delivering all of those with the Cas12a Ultra. this protein that's been evolved to have higher activity. You can see very high levels of editing, the average being up near 100%. And gray is the same sites that are in similar regions delivered with Cas9. And so now you can see that Cas12a and Cas9 work both very well, very similarly have high activity. So how do you choose which one to use? They both do work extremely well now, especially using Ultra. So really what we find driving our decision is homology-directed repair, when you're going in and correcting that gene and looking to see where you want to make that correction. If there's a Cas9 PAM site, so an NGG, or a Cas12a, a TTTV, which has a PAM site closer to your edit. Because we found with doing HDR, the closer your edit is to that cut site, the higher level of HDR you're likely to have. It's very important to have a guide RNA and protein that's going to give you high levels of cutting, but then you want to be as close also as you can. So that, kind of helps drive which one we'll choose in our lab to work with. And then also our Cas12a Ultra protein works well at low temperatures. So this has been really great for people working in plants or zebrafish where you want to do the cutting at something more like 25 degrees. So that's a lot of the work we've done to evolve the proteins. Obviously, we make nucleic acids, so we've done a lot of work over the last 5 years to develop really efficient guide RNAs as well for Cas9 and Cas12a. And so as I mentioned, the Cas9 system naturally is a two-part system with the cRNA and tracer RNA that are then can be annealed together. So what you can do is the target-specific region will be in your cRNA, so this is a short oligo that we can make in just a couple days, and we have our design algorithm to support getting your designs for these. And then you order your tracer RNA in bulk, and you can just anneal that together. And we've done a lot of work, as Garrett discussed, with chemical modifications. So all of these are going to be chemically modified in different degrees to make these stable when you're delivering them into your cell. And so the standard two-part is going to have minimal modification. This is going to be the most cost-effective way to do big library screens. Then this is going to work great if you're delivering the Cas9 as a protein, where it's going to go in and make the double-stranded break right away, where it's not needing to wait for that Cas9 to be translated, or into cells that express Cas9. We've then also done some development to come up with a more modified two-part system. So we add in some additional chemical modifications into that RNA to make these more stable if you're delivering the Cas9 as a plasmid or as an mRNA, so that as that protein is needing to be made, those RNAs are a little bit more protected. And then we also have a single guide RNA. And so as I mentioned, this is combining that cRNA and tracer RNA into a single molecule. So now this ends up being a 100 base RNA molecule. And these are also chemically modified. And I'll show some data here in the next few slides, but these are going to work greatest when you're delivering Cas9 mRNA, because these are going to be more stable in the cell with only having two ends that would be susceptible to being degraded by an exonuclease, as well as having chemical modifications in here. And then, as I mentioned, the Cas12a is already a single guide RNA, so we have that, and these are all chemically modified. These are all the options that can be directly ordered on our web, and these all are provided to you in just a couple of days. So I just briefly wanted to touch about how to make the RNP. This is these all come experiment-ready reagents. The RNP, obviously, or the protein, obviously you can have stocked in your lab. To make the two-part, you would get your target-specific cRNA. You can use the design tool to design that. Anneal that to the tracer RNA, heat that up to 95 degrees, slowly cool, and then combine that with the protein. You can do that at room temp, 37 degrees. That forms that RNP complex. And then that complex is actually quite stable. You can then directly deliver that into your cells by microinjection, electroporation, lipid, or you can actually store that in the freezer, take it out, use it again if you're working with the same target site. And then I just wanted to touch on, if you have an sgRNA, same sort of thing. You're just now not needing to anneal these two strands because we've chemically synthesized that together. And then it's the same approach with if you're using Cas12a. You're just going to incubate that cRNA with the protein and then deliver that RNP into the cell. So what IDT has been able to do is really do a lot of R&D to provide the highest quality RNA reagents that are going to have a design algorithm that's backing them up, have 3 years of manufacturing experience to have these be, of the, like Bob spoke of, the highest quality oligo with, great coupling efficiencies. And we're able now, we've spent a lot of effort over the last year and 2 years to be able to really be able to make long RNAs efficiently too. So this is great for a lot of you that are working with lipid nanoparticle delivery of mRNAs, where I'll show here the next slide how using Cas9 using single guide RNAs really works great when you're delivering with mRNA. So we're really at a point where we can make single guide RNAs research grade very quickly. All of these have those chemical modifications that I've discussed. And so to see some activity differences of the different guide RNA forms, here we've looked at 12 different sites, and you can see in the top left corner, we're comparing 12 different sites that were synthesized as our standard 2-part, the 2-part XT, which is going to have more of that chemical modification, or a single guide RNA. So in this first panel, we've co-delivered these as RNP at a 4.8 micromolar dose. And so if you focus on the white dot there as the average, you can see that all of those, when delivered with protein, give pretty similar levels of editing in this cell type. We're working in HEC293, but a little higher editing with single guide RNAs overall. And when we drop the dose down in the second panel, very similar again across the 3 different guide RNAs. But what stands out in the bottom is if you're delivering Cas9 in an expressed form, as mRNA, for example, you see a much greater advantage when you're delivering these as a single guide RNA. The standard 2-part isn't sufficient for that. So looking into more detail, as I mentioned, we've over the years built some great methodologies for doing high throughput screens. So that's all supporting our dicer substrate algorithms and our now guide RNA algorithms to do all the design work. We wanted to look a little bit more at many sites and see if there's a difference between 2-part and single guide RNAs when these are delivered as RNP. So here, this is just showing the amount of data we're able to get. And we're looking at 2-part versus single guide RNAs delivered as an RNP in Jurkat cells. And you can see very similar levels of editing in that cell type. However, we've done the same thing, taken the same 255 sites that were randomly selected and delivered those as both a 2-part and single guide RNA into HAP1 cells. And in that case, in that cell type, single guide RNAs do perform better. There are some cases where we have great correlation, but there are some sites where the single guide RNA works better. So this is the sort of thing that, we can take this data and work on building algorithms of when you would maybe use one over the other, but wanted to highlight that data here. And then looking more at sgRNAs delivering with mRNA, this is a paper that came out from Intellia Therapeutics, where they're looking at lipid nanoparticle delivery and adding in additional chemical modifications to that single guide RNA to further stabilize this when you're delivering with mRNA. And so the standard sgRNA modification pattern has 3 2'-methyl methyl bases and 3 phosphorylthioate linkages on each end of that 100 base RNA molecule. And that's what's available ordered on our website directly when you would enter in your 20 base sequence. This is what would be made. However, IDT is able to make a multitude of different chemical modification patterns, like Garrett spoke to the different modifications that we can do. And so this is a sequence that is commonly used with lipid nanoparticle delivery. And what this has now is those red bases or 2' methyl bases. So now we've further protected those loops in that sequence. And so this was published, and I just wanted to touch on this is something that we can make. It's not directly orderable on our website now, but this is something that you can get through our support team. And so to show data with that, here again, looking at taking 12 different sites and delivering them with mRNA now as a standard 2-part, not much activity, as a more modified 2-part, as a single guide RNA, which I had already showed had the highest activity, but now even going further and modifying that with modifying about half of those bases in the single guide RNA with 2' methyl bases, you can see that's where you get the greatest level of editing, both with 293 cells and in K562 cells. Our standard single guide RNAs are going to be standard desalt purified. And so here is looking at a single channel capillary electrophoresis where we're able to visualize single base resolution and get you an idea of what the purity looks like. So this is going to be for those screening, research grade, looking at identifying your sites. And so this is actually really great because we get this question a lot of, should I be using standard desalt? At what level do I switch to HPLC? And I just wanted to let you know that we can do both. Again, the standard reagent you would get on our website is standard desalt, but we can also offer HPLC purification of single guide RNAs, which is going to get you to then near 100% full-length product. And then to look at some data of how these would perform, here we've delivered the standard desalt in blue, and then the HPLC purified sgRNA in orange, and done a dose response of RNP delivery. And it's not until you get really into these low doses that you see a slight difference, again, because you don't have as much full-length material in there. But that standard desalt works very great, and this is really great screening reagent. And then along the lines of what IDT is able to offer for you, there was a new big publication that came out this fall in Nature, primetidine. I'm not sure if anyone is working on primetidine yet, but this is essentially taking a Cas9 nickase now, which is fused to a reverse transcriptase, and then it uses an RNA construct that has the target specific region and then the Cas9 recognition sequence, but now it's got the material that you want to correct. It's going to have the reverse transcriptase binding domain, a primer binding domain, and then it's going to have your correction material. So it's basically fusing the donor template with the guide RNA and delivering this all in one with a reverse transcriptase and nickase. But what I wanted to get across here then is, so this ends up needing is 100 base sgRNA like you would typically use, but now you've got additional sequences on there. So you're looking at an RNA that needs to be between 120 it's about 120 to 160 bases are the common lengths we're getting requested for that system. So I just wanted to highlight that IDT can also make those, even though that's not a part of the standard sgRNA offering. That's something that we can also support. So reach out to us if you're wanting to look into those studies. And this is all really new. We are gearing up to do some of this work in our lab and see how this technology works, and if we want to move forward and provide design sets for this, see if it works with mRNA, maybe protein. But right now, the publication delivered this all in a plasmid construct. But as I mentioned, we want to look at this with chemically synthesized RNAs. So this is just data showing the level of primetidine that they saw in the paper with the plasmid, but then now if you deliver these with a plasmid and an unmodified or the chemically modified PEG RNA, we're getting those levels similar. So just showing proof of concept that this will work and we can make these reagents. So to wrap up the guide RNA portion, I just kind of wanted to get the message across that if you don't see it on our website, we likely have a way to do more unique types of modifications or purifications, so just reach out to us. But for the guide RNA support, we've got algorithms that will support the design. We've got many different scales that will come very quickly. For the screening grade, we can do libraries. If you wanted to do large knockout studies, we've made 100,000 guide RNA libraries to look at that. We can HPLC purify these. We can, like I said, support any new technology that comes up that would use an RNA guided molecule. So just the last couple slides here, I wanted to touch on HDR. So again, this is where we're going to then now provide a donor template to make a correction. Our R&D team over the last 2 years has really put a lot of effort into trying to figure out the rules to improve HDR. Sarah spoke to, how this is much more inefficient than the non-homologous end joining. So to touch on all of this, we've got several different options to support you along the way of doing HDR. And we provide Ultramer templates, which I know Bob had on one of his first slides, but this is going to be a 200 base high quality DNA oligo that then allows you to basically insert about up to 120 bases of material with those homology arms that you're going to need. But with this, we've done a lot of work also modifying that construct and being able to provide a molecule that is going to give you higher rates of HDR. So in about a month, we are releasing what's going to be referred to as our ALTAR and modified HDR donor oligo. And I'll show some data on the next slide, but this is going to add additional modifications on the end of that ultramer that will protect this as well, like we've done with the guide RNAs. We also offer megamers, which will get you lengths up to 2 kb, and then we've got double-stranded DNA options. But we've developed a lot of data to support rules of how to help design HDR. So we've looked at homology arm lengths, the distance from the PAM site that you need to be, the effects of adding in silent mutations, using the Cas9 nickase. And so we've got a lot of data supporting the best way to do this. And so what we've really worked hard over the last year is building this data also to a design tool. And so in about a month, we'll have an HDR design tool on our website that will take all this empirical data that we've generated and help you provide the HDR donor construct along with your guide RNA that will help, that will be predicted to give you the highest rates of HDR and then allow you to get those reagents. And then the other thing that we've done work on in the HDR space is looked at small molecules that could be added in to the cells to increase rates of HDR. And so we've got a compound available right now, and we've been looking at additional ones that work across a broader cell type, and that'll be available soon as well. So this is just a snapshot of what our design tool will look like. It's going to support human, mouse, rat, zebrafish, nematode, and wild-type Cas9 as well as nickase to begin with. The Cas12a is still more hand-designed, but if you're going down that route, let us know. We have design rules for that as well. And this will have the ability to add in silent mutations, like I said, but we've our bioinformatics team has developed a great algorithm that now our web development team has made this pretty user interface to be able to provide to our customers. And so to touch the last bit of data is the modifications we've made to these HDR donor oligos, along with our HDR enhancer, now able to give you the best rates of HDR. And these are additive. So looking at 3 different sites here, if you just use an unmodified donor oligo to make a single base change in this case, and then add in our HDR enhancer, you can see the boost that you get. It's been published, and Garrett spoke to the benefits of adding in phosphoryl thioate linkages, but putting 2 phosphoryl thioate linkages on each end of your donor oligo gives a boost. And so you can see that's where you go from blue to gray here. And then now adding in our HDR enhancer. So you see that additive effect of protecting the donor oligo, as well as adding in that HDR enhancer. And then lastly is the modification that we've studied. We've looked at many different modification patterns, and this is the one that's given us the highest rates of HDR. You can now see in all of these sites with our N modification and HDR enhancer, now you're getting up to really high rates of HDR compared to where you started by just introducing a unmodified DNA oligo. And so as we're developing these reagents, we have a lot of collaborative efforts working with other institutions to make sure this works across different systems. And so we worked with a group at the Jackson Labs in Connecticut, and they published a methods paper where they're working in iPSCs. And so they have a paper now showing, using the ALTAR modified donor template and enhancer, that they've also been able to see this highest rates of HDR in stem cells. And so that paper is there, but this is showing again the similar figure that I just showed on the last slide, but now we're looking at they're doing a BFP to GFP conversion assay. So we've got some FACS data here where the level of HDR is shown up in this top panel. So with an unmodified donor template, they're at 34%. They add in the HDR enhancer, still with an unmodified donor template, they've boosted that about 1.5 times. And then they use the IDT ALTAR modified, they're at about 50%. And then you can see that final panel when they're using the modified donor template with HDR enhancer now in iPSCs, they're getting at this site about 70% HDR. To wrap this up, we really have a lot of resources on our website as well as with our support teams. So we've got protocols on our website that can support a lot of different organisms and conditions. So I encourage you to look at that and reach out to that email if there's any specific questions you have. And this is the R&D team, the functional genomics R&D team that's done most of all of this CRISPR research over the last 5 years. So thank you. And sorry for the shuffling of here.
Thank you, Ashley. Any questions for Ashley on the development of our CRISPR systems here?
I have a question related to the Cas9. So for Cas9, you offer guide RNAs as well as the protein part.
Mm-hmm.
For the prime editing, you now starting to offer the nucleic acid part. Do you plan to offer the protein part?
Well, we have to test that first and see if that works. I have not seen any data yet of anyone trying that with protein. It's all been plasmid and mRNA, but we're investigating to see if that's a feasible option.